Now let's finish the 12th chapter of biology for the MCAT, which is the last one, and we're done with biology finally. So let's do it and get through it quickly. Okay, so we have several definitions here, and these are genetic definitions. Then we are going to learn about the Mendel's laws, two laws of segregation and independent assortment. Then we have a bunch of experiments: the Griffith, Avery, MacLeod, McCarthy experiment, and Hershey Chase experiment. Then we have nucleotide mutations. Then we have chromosomal mutations. Then we have analytical techniques, and then we have evolution. Okay, now let's start with the definitions. So, what is an allele? Alleles are alternative forms of a gene. So, the dominant allele only requires one copy in order to be expressed. Recessive allele requires two copies in order to be expressed. So we all have gene expressions. And in order to be expressed, it means that your genes are going to be translated into, so your genes are going to be transcribed into an mRNA, and then the mRNA is going to be translated in order to be expressed and give you the necessary proteins that gene codes for. Then we have genotype, the combination of alleles one has at a given locus. So we need to have an understanding of locus as well. I believe locus is that local gene, right? So the combination of the genes, the alleles, is a genotype. So at a given location, specific location on your genes, or basically your chromosome, you have genotype. The combinations of those alleles are called genotype. We have homozygous, which is two of the same alleles. So a homozygous recessive allele will be expressed. And then we have heterozygous, which is having two different alleles. Then we have phenotype, which is the observable manifestation of a genotype. So this phenotype is basically the characteristic that you will see from that genotype. So you will see the characteristics and traits in your body, for example. This is what you will observe, right? It's a much bigger thing. It is a superficial thing that you see on the face of someone, or a plant even, or any alive substance. So genes are, here we're talking about biology, which is life, right? Then we have dominance, so complete dominance, which is only one dominant allele, right? And then we have codominance, which is more than one dominant allele. So you have two dominant alleles, that's codominance or more. And then you have incomplete dominance, which is no dominant alleles. And heterozygotes have intermediate phenotypes. So what does that mean? If no dominant alleles, then it's two recessive alleles, for example, right? Or three recessive alleles, right? And you call this a incomplete dominance. And the heterozygotes have intermediate phenotypes, whatever that means. We'll figure that out. Penetrance. So intermediate phenotypes, for example, the intermediate phenotype of a red flower and a white flower would be pink, right? something like that. It's intermediate. So if the red is a recessive allele and the white is also a recessive allele, then the incomplete dominance would be a pink phenotype. Then we have penetrance, which is the proportion of individuals carrying a particular allele that also express an associated phenotype. So the proportion of individuals carrying a particular allele, for example, a red flower, expresses an associated phenotype. So the proportion is the penetrance. So how many red flowers do you see in compared to the general population of the flowers? Expressivity, which is the varying phenotypic outcomes of a genotype. You have the white flower, the pink flower, the red flower. These are different expressivities, the varying phenotypic outcomes of a genotype. So one genotype, for example, a heterozygous genotype, is giving you expressivity. So some of the heterozygous phenotypes are orange, genotypes are orange, and some of like varying phenotypic outcomes, right? Some of them are orange, some of them are, I don't know, like a shade of light red, right? A shade of a dark yellow, stuff like that. Then we have genetic leakage, which is the flow of genes between species via hybrid offspring. We have to see what is the hybrid offspring. But the flow of genes between these species, different species, with hybridization. For example, you see, probably this is like an example of a zebra, which is, I think, the mix of horse and donkey and an Oreo. I'm joking, but you get what I'm talking about, right? So this is genetic leakage. That's interesting. So hybridization is associated with genetic leakage. The leakage might give you the another common sense on meaning. Like for example, you might think that, like right now, I'm thinking that when the genes are leaking, when something is leaking, you are basically losing it, right? Uh, However, this is not in the case of genetics. This is not losing your genes. This is when you get the zebra, as I explained, right? Then we have genetic drift, which is when the composition of the gene pool changes as a result of chance. So the composition of the gene pool, so all these genes you have, right? The composition of them changes as a result of chance. So, for example, yeah, we will get the examples later, but just for now, drift is the result of chance changing the genetic pool, the composition of genetic pool. Then we have also the founder effect, which is bottlenecks that suddenly isolate a small population in breeding. So we have suddenly so you can suddenly isolate a small population, and this is called inbreeding. So a specific breed, of the animals, for example, birds, for example. So only white hawks will be breeding with each other, and then that's when you get the founder effect. Now, the founder would again might mislead you into thinking that those those genotypes are giving phenotypes that are the founders of that genotype, right? Something like that, but that's not the case. We have inbreeding is the founder effect. So white hawks with white hawks, for example, giving more population of the white hawks in general. Now we have taxonomic rank. So kingdom, phylum, class, order, family, genus, species. Now this taxonomic rank I have not fully understood yet. Let's actually dig into it and explore it together. But we have King Philip came over from Great Spain. King Philip came over from Great Spain. K for kingdom, Philip for phylum, however you say it. Came is class, over is so I'm not reading it. I'm just trying to over the O order. Okay, order from family, right? Yes. Great Spain, G great genus, and Spain species. That's good. That's good. Okay, Mendel's laws. We have law of segregation, which is when an organism has two alleles for each gene, which segregate during anaphase 1. So we have segregation that happens during anaphase 1. So PMAT, if you remember, right? And the anaphase was the third stage of meiosis, right? Of one, right? And the during anaphase, the separation happens. So during metaphase, which is the stage before that, the chromosomes or sister chromatids meet in the middle. So for meiosis 1, we have the homologous chromosomes that meet in the middle for metaphase 1, and then in anaphase 1 they are being separated. So the is it the mitotic spindles? Those are detaching, separating away the homologous chromosomes, and because of this, so the law of segregation. So those two alleles for each gene segregate during anaphase 1. So one of them goes to one gamete, the other one goes to the other one. Because of this, gametes carry only one allele for a trait. So what does that mean? You bring so each gamete has one allele for a trait, not two. Right? You only have long hair, not short hair. That's the law of segregation. The other one, law of independent assortment, is the inheritance of one allele that does not influence the probability of inheriting a given allele for a different trait, except for linked genes. Interesting. We'll dig into that, but I kind of understand this right now. When you inherit, one allele is not having an effect on another allele, and its inheritance does not influence its probability. Probably this relates to the Hardy-Weinberg equation as well. That's when you are having the independent assortment, right? And there's an exception, which is the linked genes. It means that these two genes are actually linked together. That's why this happens. Now let's go to the experiments to support DNA as genetic material. We have the Griffith experiment, which was demonstrated by transformation. I think this was also in the first chapter, interestingly. And heat killed smooth virulent strain of bacteria, still transformed rough strain into smooth. So I have a screenshot of this and I put it in the transformation slide on the Anki deck, and I can share that with you later. But the Griffith experiment basically demonstrated the transformation, okay? And this transformation is basically the so from so this is fresh DNA from the environment, right? This is how we know of transformation. So the heat killed smooth strain of bacteria, which is virulent. It has the viruses, right? Still transformed rough strain into smooth, right? So it still made the other one bacteria, the rough strain one. The rough one was safe, right? And the smooth one was virulent. But however, the heat killed by itself did not make the bacteria dangerous, deadly, but still, through transformation, the DNA was from the environment was transformed into the other bacteria that was safe and the rough strain, and it transformed it into smooth. Now we have the Avery Macleod. How do you say this?
McLeod.
McLeod. I'm so sorry. I don't know why I was pronouncing this differently. My English sucks. So Avery, McLeod, McCarty. These are three scientists, as we can see by their name, and they so degradation of DNA led to a cessation of bacterial transformation. Degradation of proteins did not. So when DNA was degraded, it led to a cessation of bacterial transformation. So bacterial transformation did not happen anymore. So these three scientists, just by this statement, we assume that these were after Griffith, not before him, right? And because they already had a knowledge of transformation, and they concluded that if you degrade DNA, that will stop the bacterial transformation. But if you degraded proteins, it did not. Now, it's almost the same as Griffith, right? Because he killed it, and he does kill proteins, but the DNA did not break down, right? Now we have Hershey Chase, which who confirmed. So these are two scientists. So we go 1-3-2. One scientist is Griffith, then we have Avery, McLeod, McCarthy, and then Hershey Chase, who confirmed that genetic material DNA is genetic material because only radio-labeled DNA could be found in bacteriophage-infected bacteria. Interesting. So they labeled the DNAs, and only DNA could. So the radio-labeled everything basically, I think, and the radio-labeled DNA could be only found in bacteriophage-infected bacteria. So bacteriophage was these specific viruses that infect bacteria, and they have the tail sheath, and they inject their viral DNA into the bacteria. And when they radio labeled it, they saw that only the DNA is transformed into the infected bacteria. Now we'll come to the evolution in a moment, but here we can see that we have divergent evolution, a parallel evolution, and convergent evolution. Now, for mutations, we have the nucleotide and chromosomal mutation. For nucleotide, we have point mutation, which is substituting of one nucleotide for another. So A for C, maybe, or T, whatever. And we have frame shift mutations, moving the three-letter reading frame. So frame is for the three-letter. The whole, you know that we read the codons 3 by 3, and each 3 gives for a protein. So yeah, that's the frame, right? Frame mutation. But only one nucleotide, that's point mutation. And the results however, it could be silent, having no effect on the protein, because we have what did you call that? Wobbles? What, like whatever that the codons basically, right? Yeah, the wobble. That was a wobble. So on the third one, you can actually change the nucleotides and have a silent mutation. And then we have missense and nonsense. So what's the difference between miss and non? Missing something is replacing one amino acid with another, so you missed it, right? But non is non-stop. You can take it like that, think it like that. So it's non-stop, which means this is a stop codon. A stop codon replaces an amino acid. And insertion, deletion, I'll just put non-stop for mnemonic sake. For mnemonic sakes, I'll I just put it there. Then we have insertion, deletion, shift in the reading frame, leading to a change in all downstream amino acids. So when you shift in the reading frame, right, it leads to a change in all downstream amino acids. So that's either insertion or deletion. You add an amino acid, you add a reading frame, or you delete one. And that causes a downstream mutation in the amino acid. Then we have chromosomal mutations, which are much larger mutations affecting whole segments of DNA. This results in deletion, duplication, inversion, insertion, and translocation. So deletion is, as the name applies, a large segment of DNA is lost. So you lose a large DNA segment. Duplication, you duplicate the DNA segment multiple times. Inversion, the segment of the DNA is inverted, reversed. Insertion is a segment of DNA which is moved from one chromosome to another. So you have a chromosome here, and you move that segment of DNA into another one, and not to be confused with the nucleotide insertion mutation, which is a shift in the reading frame that causes downstream amino acid changing. But the insertion of chromosomal is when one gene goes from one segment of DNA is moved from one chromosome to another one, right? And we have translocation, which is a segment of DNA. They swapped with a segment of DNA from another chromosome. So there's a difference between insertion and translocation mutation of the chromosomes. And for the insertion, just one segment of DNA goes to another one, but for translocation that DNA segment is also swapped. So the DNA segment that the previous DNA segment goes to, that replaces the one that it just left. I'm just trying to play with words to make myself understand it better. Then we have analytical techniques. So we have the Punnett square, which is a monohybrid cross that accounts for one gene. So when you hybrid crisscross basically, and the mono applies for one allele and with another allele, and it accounts for one gene. Then we have hybrid crosses that account for two genes. And we have sex-linked cross that is linked to the X chromosome. Then we have recombination frequency, which is the likelihood of two alleles being separated during crossing over in meiosis. The father is more likely, whatever that means. Does it have to do anything with the XY chromosome? Probably. So the probability of two alleles being separated during crossing over in meiosis, this recombination frequency, and recombination if you remember, was related to um, those two chromosomes, the chromatids having a chiasma and recombinating, basically crossing over each other and exchanging their DNAs. Then we have Hardy-Weinberg principle, which is if a population meets certain criteria aimed at a lack of evolution, then the allele frequencies will remain constant. So if a population meets certain criteria aimed at a lack of evolution, then the allele frequencies will remain constant. So basically, there's going to be no more recombination frequency, right? Okay, I'm gonna highlight these tricky ones, right? Genetic drift, genetic leakage, like the zebra. We have expressivity, we have penetrance, we have taxonomic rank, we have law of segregation, independent assortment, the Hardy-Weinberg principle, again, is when you have no longer the recombination frequency. And then we have the equation for Hardy-Weinberg, which is P plus Q equals 1. There's going to be questions on this that we can just solve, and we can solve it together. And then P squared plus 2PQ plus Q squared equals 1. And that's about that. Then we have the evolution, which is natural selection. The mechanism for evolution is natural selection. So the mechanism of evolution is natural selection. We have modern synthesis model, which is neo-Darwinism. Mutation and recombination are mechanisms of variation. Differential reproduction. So neo-Darwinism. Mutation and recombination are mechanisms of variation. Differential reproduction. We have inclusive fitness. If a population meets certain criteria aimed at a lack of evolution, then the allele frequencies will remain constant. Then we have punctuated equilibrium. Considers evolution to be a very slow process with intermittent rapid bursts of evolutionary activity. Punctuated equilibrium. What does punctuated mean? You mean you're punctual, being precise, right? Brought to a point. Punctuate. Put dot, point, accentuate, break up. Slides were used to punctuate the talk. It's very slow with intermittent rapid bursts of evolutionary activity. So evolution, punctuated equilibrium theory probably is the theory, states that evolution can be slow, very slow, but at times there are rapid bursts that just change the whole game for evolutionary activity. Then we have mode of natural selection. We have stabilizing selection, which keeps phenotypes in a narrow range, excluding extremes. Then we have directional selection, which is moving the average phenotype toward an extreme. Then we have disruptive. So average phenotype towards an extreme is directional toward excluding extremes, stabilizing. Disruptive, moving toward two different phenotypes at the extremes can lead to speciation. So moving to two different. The key for disruptive is two. Adaptive radiation is rapid emergence of multiple species from a common ancestor. Each has a niche. Do we have niche here? No. So I thought niche is a business terminology, but a comfortable or suitable position in life or employment, a specialized segment of the market for a particular kind of product or service. Yes, that's the niche that I know. Everyone knows usually. And denoting products, services, or interests that appeal to a small specialized section of the population as an adjective. Other companies in this space had to adapt to being niche players. And then we have as a verb, place something in a niche or recess. These elements were niched within the shadowy reaches. Yeah, whatever that means. It's from early 17th century, from French literally recess, from nicher, make a nest, based on Latin nidus, nest, recess, alcove, nook, cranny, slot, slit, hollow, bay, cavity, cubbyhole, pigeonhole, opening, aperture, mihrab in Islam. And he feels he has found his niche in life, ideal position, calling, vocation, métier, place, function, job, slot, opportunity. Okay. Then we have isolation, which is reproductively isolated from each other by pre or post zygotic mechanisms. Geez, what are these? But isolation is just like reproductively isolated from each other by pre or post zygotic mechanisms. Zygotic, like spermozygotes we have, right? We have eggs also as female zygotes, stuff like that. So, reproductively isolated from each other by pre or post-zygotic mechanisms, whatever that means for isolation. Then we have molecular clock model, which is the degree of difference in the genome between two species that is related to the amount of time since the two species broke off from a common ancestor. The molecular clock model, the degree of difference in the genome between two species. Which is related to the amount of time since two species broke off from a common ancestor. Molecular clock. Yeah, I didn't know. I'm working right now on a project of biological clock, and yeah, we have a molecular clock as well. Interesting. And these are the three different types of evolution. Okay, let's go see how many cards we have. So just for that, let's save the edits. Then let's see how many cards we have for chapter 12. We have 101. Interesting. Let's unlock them. Let's now go to the biology deck and rename. Oh, sorry, on my other screen. Yeah, we don't want this. We want custom study by one. Then it was 102. Let's put one more. Choose tags, chapter 12, genetics. We don't want anything else selected. Great. We okay that. We have 87 to learn. And because some of them are duplicates, probably. And we name it as B12 genetics. And let's take a look at our deck here. Let's rename these actually. B7 cardiovascular. Just add the B before it. This goes B8 immune system. This goes B9 digestive system. And we are good to go, I think. So we have B1, 2, I don't know why these are here in the middle. But, oh, because it has the number after it. B10, B9. Can we now replace it? Bring it. Okay, we just want to bring it before it. Let's see. Can we bio dex? Now let's add them here. Where did it go? Where did you go? Okay. I don't think that was a good idea. B1. Whatever, I don't know why. Yeah. Okay, I'll see you guys later. I'll figure this out and just take a short break and then we'll be back for the cards. Peace.